I'd like to briefly introduce you to the genetics analysis module of Savi tools. In this video tutorial, I will guide you through some of the functions and analysis capabilities offered in the module. Here you select Genetic Analysis, and this will direct you to the module home screen. This application provides example data that is already uploaded and ready to analyze. Alternatively, you can upload your own data. You can select either of these options in the left-hand panel, or you can download example files that provide an example of the required formats. Here we will use example data, and this includes a multiple sequence alignment FASTA file of OR5 gene sequences from porcine respiratory and reproductive syndrome viruses. We also have an optional attributes file. This assigns categorical data to the sequences. In this example, it includes 3 attributes, including RFLP type, the farm system, and the year of isolation. Again, including attributes is optional. However, downstream, it will allow you to filter your data by specific attributes of interest and visualize the phylogenetic relationships by these attributes. We click Explore Outputs, and the first screen displays the distance matrix. This can be viewed in a customizable matrix or as a basic table. The distance matrix shown here indicates a pairwise genetic distance between sequences, showing the number of sites that differ between each pair of sequence. For example, 21 nucleotides differ between isolate 2528, and 6332. If you're only interested in comparing viruses isolated in 2015, for example, you can filter this matrix by year, one of the attributes, by simply deleting the years that you're not interested in viewing. You can also change the display of the matrix by specifying the upper triangle and lower triangle formats by selecting this in the drop-down menu to the left. So next we can visualize a phylogenetic tree of our data, and this can include a neighbor joining tree or a maximum likelihood tree. Here we'll explore the maximum likelihood tree. These trees are based on a model of sequence evolution, which you need to specify. A model test is automatically performed, and the most appropriate model can automatically be selected by choosing Use Best Model with Lowest AIC. Alternatively, you can specify a different model to use. In order to view the tree and the model test results, we need to first click Generate Output. You can explore the model test results by clicking on this tab. At the top of the table, it tells you which is the most appropriate substitution model based on the uploaded sequence data. For us, it's HKY with gamma rate variation, as this has the lowest AIC. More details on these values and evolutionary models can be found in the user guide, which you can download by clicking here. Now let's navigate back to the tree view. By default, an unrooted tree is displayed with the color of the tip label RFLP type and dot-only tip style. Alternatively, we could select phylogram. We can root the tree at the midpoint or any isolate that you choose to select as an outgroup. You can also change the color of the tip label to year and show the isolate ID as the tip style. We can also align the tip labels. You will usually always want to include a scale bar to show the scale of the branch lengths. You can see in this example that isolates collected in 2014 and 2015, the blue and green isolates, are more closely related to each other than they are to isolates from any other year. You can add bootstrapping, and this can take a bit of time depending on the number of replicates that are selected. So as you can see, the genetic analysis module has many features. I only had time to introduce some possibilities offered by these tools. The user guide offers more details and explanations behind these analyses.